Okay. Hello. Hello. Am I audible?
Yep.
Yeah. So
Yeah.
I think Dr. Pavitra has. Yeah. Okay. Good afternoon, all of you. So we can start now. Okay. Yes. So hello. Good evening to all. So welcome to the third panel discussion of the series as a part of CCMB Open Week 2020. Every year we celebrate the Open Day by directly interacting with general public and from all age groups and backgrounds and explain them about research activities happening in CCMB. This year, due to the COVID-19 pandemic, we have switched to this virtual platform and extended the program for a full week. This is packed with learning, competitions, and discussions with experts. So first, I would like to give some instruction for the general audience. During the session, please mute your video and audio. At the end of the session, we will ask raising your hand for question. At the time, please raise your hand, and in between, you can also put your question in YouTube comments. So from here, we can actually start our session. So today we will be having a discussion about the stem cell research and the ethics related to it. So we read a lot about stem cells in the news and about how they are showing remarkable potential in research related to clinical therapies. Today only we got a news that among the 100 most influential people that are published by Times, there is a person called Ravindra Gupta who demonstrated for the first time that HIV can be treated or possibly treated by stem cell therapy, and he actually showed it in a patient called London patient. You can search in the net also. So from this we can actually think how much important this stem cell research is right now. Still, there is a lot of misinformation circulating about how stem cell research can be used for different treatments like curing congenital disorders. And there is still a lot of ambiguity about ethics related to their usage in research. And for that purpose only, today we have gathered here. So let's discuss this with our panel members. So first of all, I would like to introduce our panel members today. So our first panel member is Dr. P. Chandrasekhar. Uh, Dr. Chandrasekhar is a principal scientist at CCMB, and his lab is currently trying to understand the factors which determines fates of stem cells. They use embryonic stem cells and transgenic mice for this research. And his expertise in this field for more than a decade actually makes him one of the best person to discuss about the scope and ethics related to stem cell research. And our next panel member is Dr. Pavitra L. Chaveli. She is also a scientist at CCMB. Her lab actually focuses on studies related to microcephaly and neural cancers. They use neuronal stem cells and brain organoids to study these. And she has worked in this field in different countries, and that makes her capable of comparing the current research scenario and regulatory policies in different developed and developing countries. I think they're not available right now. Dr. Pavitra?
Yeah.
Are you there? Yeah.
Yeah.
Yeah. So that's all about Dr. Pavitra. Our next panel member is Dr. Pravavati Devan. She is currently a Serb National Postdoc Fellow in Dr. Josna Dhawan's lab at CCMB. She works with muscle stem cells, and Dr. Prabha has previous research experience in the field of immunology and as well as cancer biology. So we will be hearing from her about mostly regenerative medicines and ethics related to it. So starting with the main topic, I would like to ask the panelist from from a very simple question like uh, what is what are actually stem cells, and they can give a brief introduction or idea to the general public about it. So we can start with Dr. Chandrasekhar.
Okay. Thank you very much. So yeah, as Meher has said that, okay, what are stem cells? Stem cells are basically those cells which can keep dividing and duplicating to give similar type of cells for a prolonged time. In some cases, they can do it forever. That is, some cells called as pluripotent stem cells or cells isolated from the embryos. They can go forever. Whereas there are some cells called adult stem cells, they can do it for certain number of times. So this is one important property, that they can perpetuate forever or for a longer time. Second is, they should also have the ability under appropriate conditions to give rise to other type of specialized cells in the body. Okay, that's another property. Now, how many types of specialized cells they can give? It depends on something called as its potency. In case of the stem cells, which we call as embryonic stem cells, they can form literally any type of cell in the body. Whereas some adult stem cells, let us say the cardiac or the blood stem cells, they can only form the stem cells of that or the cells of that particular lineage. So these are the two major properties. One, they can keep making themselves more numbers forever. And the second thing is they can give rise to specialized cells. Okay, I think
Okay.
move on to the next.
Dr. Pavitra,
Yeah,
would you
so
like
I'll
to
take
add
over
anything?
from so I'll take over from what CSPS said. So to just give you a real life example, all of us started in our mummy's tummy from being a single cell, right? That single cell divided into 2 and 4 and 32, 16 and 32, and that's how that gives rise to fetus and then a complex adult human being as we see. Now, human beings have multiple different types of cells. So, but still, it is possible to get each of these organs from one particular stem cell. So, if you give a particular recipe to the stem cell, it then decides to become a brain. If you give another different type of a recipe to the cell, it will then become heart. Okay, so like that, we have different recipes to grow each of these cells, which we in the lab rely on to make different types of organ systems or to study different types of organ system. So a simple example could be that when Chandrasekhar told about liver, about specific adult stem cells. So for instance, if there is a liver damage, so liver is one of the tissues which can regenerate itself because there are certain adult stem cells which are present in the liver which can give rise to liver again when there is a damage. It is the same thing for brain. So in brain also you have certain adult stem cells. If there is a minor repair, minor damage, then the brain tissue can repair itself and give rise to more of these tissues. So this fetal stem cells and the adult stem cells actually are what maintains the growth and death and maintain the homeostasis of any tissue or the organ system. So over to you, Suraj.
Yeah. Dr. Devan, would you like to add something to that? Sorry.
Yeah, hi. And thanks for having me here. And sort of a reiteration about what Dr. Chandrasekhar and Pavitra told. I want to add a different perspective to this. We all have got some sort of pain or injury, day in and day out, and few days later it gets vanished, right? So that's basically because of the healing that was driven by the stem cells, which we have, and especially the adult stem cells, which we all know, we can realize that they are doing their jobs. Just by recovering from the injuries. I work on muscle stem cell. So skeletal muscle is one of the major, motile organ, right? And day-to-day wear and tear would also injure them. And within few days, you're back to normal, right? So that is because the resident stem cells have got into the healing process. They resolved the injury and the function is back. So in that way, adult stem cells, you can realize, like you can see their function in your day-to-day life. And when it comes to adult stem cells, there are different, especially the mesenchymal stem cell is of, great attraction to me because of its, magical property of, bringing in healing in all different types of organs. So now as we go about, we can talk in detail. Thank you.
Yeah, sure. So this gives us a very good idea what stem cells are and what they do in our body. So as they are so important to us, and they are so important for the research world. So and again, we are also seeing a boom in the stem cell industry and the field of regenerative medicines. So why it is so important? Can you please elaborate on that? Dr. Devan, you can start with the perspective from regenerative medicine.
Right. So, as they mentioned, the adult stem cells, they are lineage determined, right? So, whenever, I mean, most of the tissues, like including your skin, hair, liver, brain, your muscle, and most of them have these resident stem cells, which are minimal number in population, right? So, whenever there is an injury or any signals of disturbance or disease condition, they get the trigger. Most of the stem cells are like quiescent in nature; they are not actively dividing. As soon as they get the cues, they express a lot of genes required for its proliferation. They clonally expand and then functionally differentiate to the organ from which they origin. I mean, the tissue from which they originate from. So that's the basic thing. And there are, when you look into the perspective of the role of adult stem cells, obviously the major significance would be in the regenerative medicine because of its reparative or the repairing properties. In those lines, mesenchymal stem cells are widely present, and you can see, I mean, the source is widely present, like bone marrow, your peripheral blood, and most of your bone marrow stromal cells, they give rise to mesenchymal stem cells. And it's a sort of they initiate and orchestrate whole of the healing process. So whenever there is an injury, the mesenchymal stem cells act like a maestro in an orchestra. They prime and recruit, or they prime the resident stem cells into progenitor cells, and in addition, they release a lot of signaling factors which also aid in the other processes like the blood vessel formation and the whole reparative, the resolution of the damage happens. And in that way, mesenchymal stem cells are really sort of injury-specific drugstores. So if you put mesenchymal stem cells in muscle, the way the number of, the molecules it's going to release is completely different from when you inject them in the lung. So according to the tissue requirement, the whole biological mediators released by them differ. And also, if you look into the clinical trial perspective in the regenerative medicine, you would find not less than 500 clinical trials registered using mesenchymal stem cells, and most of them targeted towards bone musculoskeletal disorders like arthritis or spinal cord injuries, a lot of immune-related disorders, and inflammatory disorders, and of aging-related problems. So you can say that the mesenchymal stem cells awaken the health system in addition to what the other stem cells Dr. Chandrasekhar and Pavitra would talk about, which have their role in the developmental program, and as well becoming into a specialized tissue.
Okay, that gives us a very good idea how important it is in the regenerative medicine. So, Dr. Chandrasekhar, would you like to elaborate why industries are so much interested in this field? Sorry, I think it's not on. It's muted.
Okay. So the stem cells are great tools for therapy, but therapy is, I think, will take a little more time to come because we still don't understand the cells very well. We are in the process of it, and I think it takes time to come. Currently, why industries are interested is because they work as a very good model system to mimic the human diseases or developmental processes. So that's where the major utility of the stem cells is currently. So for example, I can give you an example, like the drug screening any drug development process that happens, first they are done on the cell lines, then it goes to the animal models, and many of the drugs which clear the animal models fail to clear the clinical trials because the physiology of animal and the human is different. Whereas when you can make certain organs or certain types of tissues from the human cells, now they become a amazing tools, and they can also be made in large quantities, okay, very easily. That's one thing. Second, ethically also it is a good thing because you are not hurting the animals, you are decreasing the use of animals. Environmentally also it's beneficial, and it's very cost effective. And the scales can be humongous. Okay? The scale of operation is very high. So this becomes a very attractive tool for drug screening. So that's where one of the major utility that is being considered, and that's why a lot of companies are flocking to develop platforms as well as models of these stem cells to mimic human diseases in a plate and try to screen for the drugs as well as to understand the molecular basis of the disease. So I think we can move to Pavitra and she can probably from here.
Okay, so taking over from what Chandrasekhar said, so he was talking about how we can make mini organs, or you can make mini human replicas, basically human organ replicas on a plate. And that has been quite attractive because it is not only one particular organ, but people have been trying to do mini brains in the dish, which is what we work on in our lab. They've been trying to make mini liver, mini pancreas, anything, mini heart even. So you name it, mini ovaries. So what happens is sometimes, I would just extrapolate it a bit more and talk about diseases such as cancer. Okay, so we know that how many of our patients undergo different types of chemotherapy or different drug regimen, the cancer kind of normally relapses, or it is impossible for us to know that which particular drug will work for this particular patient. Okay, in which case these organoids are a very strong model system, because instead of taking a mouse and treating the ovary of that mouse, it is much easier to actually take the cell from this patient, make it into an organoid, okay, and see which drug this particular cancer in that particular ovary responds to. Okay, therefore you would have a very good drug screen. And like what Chandrasekhar said, you can't. It is not necessary that you would do it in one ovary. From a single cell from one patient, you can end up making close to hundreds and thousands of organoids. Okay, so that is a really powerful way to say that, okay, this drug is going to now be resistant, this cancer is resistant to this drug, or it is going to be sensitive to that particular drug. So that is a very strong plus we have. And not only these kind of mini organs, if you also think about how we can stimulate stem cells to make more. I think a lot of you would know about Michael Schumacher's skiing accident, right? So what he was put on was on a deep oxygenation regime, okay, which triggered his brain cells to trigger making more stem cells, which would actually recover his brain, right? So these kind of therapies are also in place. It is not only taking a stem cell and putting it into a patient or doing something like that, but also triggering the cells which we already have in our body to make the correct type of cells.
Yeah. So I think this gives us a very good idea that why stem cells are very important. So considering such importance of stem cells in research and clinical therapies and also in industrial applications, there still exists a debate or controversy about the use of them in moral and ethical perspective. So how can we as a researcher say or we can try to avoid that, or we can surpass that for the greater benefit?
Okay, so coming to this, I think we need to distinguish between the research and the therapy. Yeah. These are two distinct businesses, okay? So research, most of the time, utilizes cells which are drawn from certain sources, okay? And commercial implications are minimal, okay? And of course, there are cases where the human patients are also involved. This is one set of things. Therapy is a totally different ballgame, okay? And most of the controversies, if you see, are coming from the therapy. Of course, there are certain level of controversies coming from the research as well, but bulk of them come from the therapy. And the root cause for this is basically after the stem cell boom came 1.5 to 2 decades back, there were no regulatory processes available or streamlined in most of the countries. In fact, India was one of the hubs for this sort of abuse of stem cell therapy. Okay, so then what has happened is people realize this, and now very strong regimes have come out in most of the countries where there are strict rules and regulations which try to restrict the therapy unless they have gone through the clinical trials and all. So many therapies were done without clinical trials. When it comes to the research aspect, I think the bulk of the controversies are surrounded towards the embryonic stem cells because they are derived from an embryo. So are you hurting a potential life? But most of the times, the stem cells are actually derived from those embryos which normally wouldn't make a human, because these are from the low-grade embryos that come in the infertility clinics. Okay. So, of course, the due credit to the donors and these things have to be taken care. Okay. And what level of research one carries on with that is a major question. Okay. So with this sort of things, like, do you want to create or edit the cells, change the things and create a human of this kind or an organ of this kind? Okay. That's one level of thought process. Of course, it also goes into the later level that do you want to even change the genome of the people, which probably later on can be given to the germline and make a new human being. So this sort of things, there are boundaries which we will never be able to cross those. So those are pretty well said. So the current, I think the issues are about using the embryos, which to large extent are settled. That's one thing. And of course, giving the credit to the donors, that's another very important thing. Okay. I think Pavitra can take.
Yeah, Dr. Pavitra.
Yeah. So one thing about human embryonic stem cells, what Chandrasekhar said. So one way to bypass use of embryonic stem cells to make this is actually by using another slightly different stem cell called induced pluripotent stem cells. Okay. So the way normally we make these is that we take skin cells from people, from patients or healthy people, and you can reprogram these to forget the kind of cells they are and go back to being stem cells. Okay, and from these stem cells, you can now start building one particular organ or two particular organs, whatever you want. Like what I said, we can use different recipes and make them. So this has been quite lucrative, and there has been a iPSC bloom for the last 10 years. People have been trying to compare and see whether there is a difference between using an iPSC and an embryonic stem cell or not. Because with iPSC, there is, of course, you have ethical regulations to consider, but then this whole question of, philosophical question of, is it going to be a life or not? That kind of a step you kind of go back on. Okay. Nevertheless, I agree with what Chandrasekhar said, that most of the problems actually come not because of use of these, but because we edit these. Okay, and you want to correct or rectify the errors which are present already in the system. So we still don't know how these would actually react in an existing system already. Okay, so people have been practicing, they don't call it a stem cell therapy, but bone marrow transplant has been there. What we do in bone marrow transplant is basically you have those stem cells in your bone marrow. When transplanted, right, you would have all these hematopoietic stem cells in them. You would have a population of stem cells which help repair that particular individual's system. So we've been practicing it using different names, but nevertheless, now that the awareness has come, people are a bit more careful.
Okay. So would you like to give us some idea about what are the governmental policies and how these are different in different countries, like in developed or developing countries?
So regulations as such, there is no difference, except probably Scandinavian countries. The regulations elsewhere are the same. I don't see a big difference at all. Okay, the trials, whatever we go through, all the trials and tribulations are exactly the same in developed and developing countries. There is no difference. And if you were to think of India as a developing country, I don't agree, especially on this policies. Yes, it was the case probably 10 years ago, but now all rules, and so we have something called good lab practice, which they really take into consideration. And then if you're making it into an industrial scale, they have something called as good manufacturing practice, okay, for people who are doing it in a bulk scale. So all these rules and regulations are present. So as such, I don't see a big difference.
Okay. So Dr. Devan, would you like to add something to the
Yeah, I would like to add a few points on like the debates and the controversies around that. So, as I mentioned, the difficulty at both the technical and scientific level is really huge. So, it's important that there is no sort of any genomic instability over the process of culturing them or differentiating them to a particular specialized cells. So, that is a major technical challenge. And in addition to that, there is a very tight ethical evaluation which scientists must ensure. As Pavitra said, like good manufacturing practices, good lab practices, and regulatory compliance, which is very important. So unless all these are met well and stringently, it's going to be, it's going to undergo controversies. And when it comes to, the ethical issues would also cover up the genomic instabilities and adverse reactions it might happen when these are not properly evaluated using stringent analytical assays or procedures. So,
okay. So coming on to the next big question. So what future do you see for stem cell research in India, and what can be done to encourage better research and also usage of these, keeping in mind the ethical concern related to this in the country like India? Dr. Chandrasekhar?
Yeah. So I think ethical issues are to good extent taken care in India right now. So at the apex, there is a NAC SCRT, which is the National Level Stem Cell Research and Therapy Committee, which formulates the guidelines. Okay. And this was jointly constituted by ICMR and DBT. And at institution levels, in case of research institute, there is something called as ICSCR, which is Institutional Committee for Stem Cell Research. And in case of clinical research, anyway, their IRBs, the internal review boards will be taking care of it in the hospitals and all. So all the stem cell research, irrespective of the source, let's say human or any animal source, an established cell line or a new cell line, everything. Now, any studies on this, be it research or therapeutics, whatever it is, has to be first going through these committees, which contains a socially aware person. Then it also contains a person from the judiciary, then a expert chairperson, and couple of experts, external experts, who will be vetting the genuinity of the proposals, and that's when you are able to conduct the research. So these things are pretty well in place and they are followed across the country now. So that way, I think the ethical issues are to large extent taken care, and mostly so. If you go back to the newspapers 10 years back and now, you will find a huge difference. There would be claims, somebody saying we treat this disease by stem cell therapy, that disease by stem cell therapy, that you don't see anymore. That's because now this has been pretty well established and streamlined in the country. So this is taken care. And coming to the opportunities, there are plenty of opportunities because we still don't have a huge critical mass in the country in any of these major fields. Okay, let's say the embryonic stem cells or even in the adult stem cells, we have only couple of good labs around the country which are doing this sort of work. And there is a huge scope for and potential for growth. And also there is a huge market also for this, both clinical as well as industry. Okay, at this point, I think I'll ask Pavitra to take it forward.
Yeah, so when he said there was a big market, actually, especially for hematological disorders, the market is actually quite huge. So hematopoietic stem cells, the corrective hematopoietic stem cells, if they can be placed in the bone marrow of patients for treatment of thalassemia, for instance, that has proven quite successful, and that is quite something which is in the market. So rest of these stem cell therapies, they are, I would say, in infancy, primarily because we still do not understand the complete complicacies and the intricacies of what these cells are capable of in doing. So when we think about research, yes, they will help us. You imagine that you have an organ drawn in a two-dimensional paper for you versus having a 3D printed organ in front of you. So that is the difference stem cell makes to the scientific researchers. So as opposed to working with cells which are just cultured on a plastic, you now start working with the organ system and something which has, an intricate architecture which closely resembles a human system. Then that gives a much bigger scope for pursuing our research, understanding biology, which in turn will help us come up with very good corrective measures or some kind of prognostic or diagnostic markers.
Okay. So sometimes it becomes very very hectic or very hard for the people to get a clearance in this type of research. So people sometimes get discouraged like young scientists, they get discouraged for this kind of efforts. So how can we encourage the young minds to get into this type of research actually?
I think this is actually quite an exciting research because you're entering into an uncharted territory. Okay. Regarding the clearances, I think if a proposal is put together really well, every place has to go through. I mean, you're working with a living system in the end. So whatever you do, it has to be done with utmost care and responsibility. And that is why these regulations exist in place first. It is ton loads of paperwork in whichever country you go to, wherever you do, whatever you want to do. As long as there is a proper scope and a good, logic to what you have proposed, I think it is a one-time thing. So you get your clearance and then you will get it renewed from your stem cell committee every year, provided you don't do. So there are 3 different kinds of modifications you can make to those stem cells. Typically working with stem cells, you will get a clearance. It shouldn't be quite a difficult process at all. But when you start making modifications to these stem cells, it could be a minimal modification which doesn't affect it a lot. Like, you want to express a particular protein, that shouldn't affect it a lot. But if you start making substantial modifications like genome editing, okay, or major changes, like you're going to delete a gene and you're going to make it into something completely different, you're going to take that organ or organoid, whatever, you're going to put it into a mouse. These are all substantial things which, of course, they will be difficult to manipulate, but we should realize that with great power comes great responsibility, right?
Yeah.
So I would like to add here one thing. You asked that how to make it attractive for students to get into stem cell research. I think one of the main attractive thing about stem cell research has been that it has never been the same. Every 2-3 years, you see some new breakthroughs coming, a new tool coming, and surprises that you think something is not possible will start happening in front of you. Now, I think one of the areas I can just tell you is when we thought, okay, stem cell biology is slowing down, now people have made embryo-like structures which can even implant and induce pregnancy. That means we are talking about the stem cells having the memory to form the embryo. And so these things have happened in the past 2-3 years. So this is what you would see if you look at the history of the stem cells, that every few years there are some exciting things that come and give you a new direction. So this is an ever-changing field. So that's one thing. If you are really adventurous and you want to do new things, this is definitely the place. The reason is you don't know what is there in next 2-3 years coming in front of you and what surprises are waiting. So I think that makes it a really exciting field, particularly for youngsters who are adventurous to venture in.
Yeah. So Dr. Devan, as an active researcher, what would you like to add into this?
Yeah, I would like to add 1 or 2 interesting aspects of stem cell research, why that should be nurtured more in India. One, which in the opening statement, Dr. Chandrasekhar mentioned about in vitro disease modeling, which is really a great sector to invest on. And this is going to aid the drug discovery because cells which are not, for which the source is not accessible, like the brain, heart, which Pavitra mentioned, where you can make organoids and try to do that drug testing for safety and toxicity and propel them for a potential therapeutics. And I mean, such things are really interesting, and there are several such sectors on which stem cell research can develop in India. And regarding the regulation part and the ethical consideration, getting acceptance for stem cell research and all, the national guidelines have clearly mentioned about like what are the different levels of studies that we are going to take forward, like ethical consideration, and as Pavitra mentioned, different levels of manipulation and different categorization of research. Everything is clearly mentioned, except for a law. I think everything is in place in India for stem cell research.
Okay, that gives us lots of information about stem cell and its research ethics. And we have lots of questions coming in YouTube comments also that I would like to convey into this discussion. So there is a question from Quick Biology. So do stem cells used in transplantation get graft rejection?
You can answer.
I think Pavitra, you can answer. If sort of a reiteration, it's okay then. No problem. You can go on.
Typically not, actually.
Okay.
Okay, just a very simple answer.
Yeah, the thing you need to take care is about the autologous and allogenicity, especially with mesenchymal stem cells. It's much easier, or you need to do the MHC mismatch or matching studies. So those are pretty easy, and that's not a huge obstacle for now. Yeah.
Okay.
And the disadvantage, mostly it is done in an autologous way. You take the cells from the patient, you culture, propagate them, whatever. In case of gene manipulation, you do that and reintroduce them, transplant them. So it just takes a lot of tedious procedure and much care to take care that the cells don't change their phenotypic and genotypic characteristics. That's a huge challenge. Otherwise, more or less, if you're done with the propagation and the scaling up, and rest all would be easier.
Okay.
I think so. Dr. Chandrasekhar cannot. No. You're on
mute. Dr. Chandrasekhar, please unmute yourself.
So I think whatever she was telling, we need to distinguish between the model organisms and the human. So in case of human, if you have to culture the cells outside and then transplant, it's altogether a different ballgame. It is treated as a new drug. Okay. So most of the times, autologous transfers or the non-autologous transfers, where the cells are taken without any culturing, they are separated and injected, that is fine. Okay. If you do a culture, it is considered to be a drug. It's a different ballgame. It goes through a different process altogether. The regulatory processes are different. Okay.
Yeah. So typically these are done when you take the bone marrow, you just flush it out and you do everything and you just re-inject. So you don't culture them outside. If you do that, like what he said, it is considered a different process altogether.
Yeah. So our next question is from Pawan. Is differentiation of stem cells are based on epigenetics?
What is not based on epigenetics? Yes, it is. So it is like this. Let's say the totipotent or the pluripotent stem cells are like a clean slate. Okay. Where if we talk about the epigenetics, the whole DNA is open for business. Most of the genes can be switched on. As the differentiation happens, it is like you're traveling in a road. There comes a bifurcation. You take one road, the other one you can't take. So that will be shut down. That's exactly what happens. So when the cells start moving into different directions, they shut down certain parts of the genome so that they don't get reactivated and go back to their previous memory. Okay. So it is epigenetic change. And exactly what Pavitra was telling, the iPSCs or the reprogramming is exactly reversal of this epigenetic process. This is a major control.
Okay.
If somebody wants to add more things, please.
Okay. So we can move on to the next question then. So Sudeep Dey is asking, do stem cell transplants can form cancer in future?
Yes, it depends on which stem cells you want to transplant. So that's why the embryonic stem cells are never transplanted. Okay. So most of the times when stem cells are transplanted, they are at a stage that they can only form only few types of cells, or sometimes they are differentiated to a terminal type of cell type and only those are transplanted. You can't transplant stem cells with higher potency in a wrong place because they can become cancerous.
Absolutely. Yeah, we call them as teratomas, right? They are teratogenic.
Yeah.
Okay, I think he has got his answer. Okay, so next question is from Anjana Srija. So does stem cell transplant have any side effects? So I think that's included in this previous question only. So next question is, how can stem cells be used for disease modeling? From Samik Sheikh.
Okay, so the way we can make these stem cells for disease modeling is in the lab what we routinely do. So let's just say that a particular, you find one particular mutation in a particular gene which causes a disease. Let's just say for an example, your hematological disorders, or in our case, we have neurodevelopmental disorders. So mutation in a gene can cause small brain phenotype. So what we do is basically we make that particular mutation in our stem cell line, and we see its role right from the day it is a cell to a mini brain. So you can study how this particular developmental disorder progresses, or at what stage does it affect the disease manifestation. Once you know that, then we can try to see at which step you can block it, okay, and or you can revert it, or you can correct it. So that is how we do with disease modeling. So we mimic the mutations or the deletions or whatever we see in the patients. We try to mimic that in the stem cells, and then we try to see how this particular model can develop. In case of cancers, we can take a fully grown mini brain or cells. We can mix this population with the cancer population or with that particular mutant population and see how this cancer now progresses, how fast it progresses. So this kind of disease modeling can be done using stem cells.
Okay. Navina Nanda, please turn off your video. Okay.
And with regards to the side effects, we can't really say any side effect, but it can backfire. So I came across an interesting news report in Scientific American, where it was it was mentioning about the embedded danger in stem cell therapy, where a cosmetologist tried to do an eyelid facelift or something for a lady in Beverly Hills. And after doing everything, he used a derma filler to patch it. And the derma filler had a substance which differentiates adipose to bone. And later it was found that whenever she was having an eyelid blink, she could hear a clicking sound. It has formed bones. So those are the sort of disasters that can happen if the doctors are not well informed. This is exactly an example of misrepresentation of stem cell therapy. So it's important that they're all well informed and they work in collaboration with basic research scientists. And these clinicians can do proper clinical trials instead of going for an unproven stem cell based therapy. They should do a proper clinical trials and go for this sort of treatments.
Okay. So Somen Kundu is asking, to form a complete embryo, will the stem cells from morphogens themselves? Will the stem cells form morphogens themselves, or is it provided by some other cells?
So when the embryogenesis starts from a single cell, it has all the information. So it creates all the morphogens on its own. Okay. So at every stage, there is switching on and switching off of certain genes, as well as the physical environment and the shape of the embryo. Okay, it's not just the morphogens, even the shape matters. These things, over a period of time, they create, there is a clear-cut program, and these are secreted from the embryo itself, and they can develop. So they don't really need external assistance as long as they have enough energy being given to them and the raw materials like the amino acids and the lipids that are available to them. And the other thing that is, in the recent times, people are trying to make embryo-like structures from the stem cells. Here it is becoming evident that certain stem cells remember, and they do have the memory of what they become or what they are supposed to become. So when people put 2-3 types of early embryonic stem cells together, they actually recognize the space and they will go and sit in the appropriate place and they try to organize themselves. That means there is an internal information that is built in. That is what actually directs them to organize into a proper embryo.
Okay.
Yeah, I would like to add a point.
Yeah.
It is not exactly morphogens when it comes to regenerative aspect of therapies. Mesenchymal stem cells and for that matter muscle stem cell, which we are studying in our lab, they do secrete a lot of signaling molecules including morphogens in something called the extracellular vesicles or exosomes. So those are body's own nano shuttles which are made, which are made through the through a very specific endocytic process, and those are packed with very essential cargoes and sent out for the purpose of communication. So this is sort of contributing for the long range signaling, which also includes, there are reports that they do carry a lot of morphogens. And yeah, it is possible that stem cells, resident stem cells, this is not like embryonic stem cell. Chandrasekhar mentioned about that. This is regarding the adult stem cell. They do communicate by releasing morphogen-like factors.
Okay. So Shivam Dogra is asking, can the growth factor that are used as transcription factors by the stem cells can be directly introduced into the body so that the body's own stem cells can grow? Dr. Pavitra, would you like to answer?
Yeah, so typically not. Because the reason why we introduce these into stem cells, okay, and make them into whatever we want them to do is because of the accessibility and the availability of these proteins. And the proteins which we are using to reprogram them, we know that they are all oncogenic. They have the oncogenic potential. Okay, so if you keep injecting huge levels of the 4 different factors which we normally use to reprogram our skin cells into stem cells, then they are shown to be oncogenic because they will induce your de-differentiation. So from being a differentiated place, cells, it will become de-differentiated. And that is the state of not only human embryonic stem cell or a stem cell, it also is the characteristic of a cancer cell, right? Because it has a de-differentiated signature. So no, you cannot do that.
Okay. So our next question is from Pujita. Can stem cells from umbilical cord collected after a baby's birth be used to treat any diseases the baby might suffer after growing up?
So yeah, Prabha.
No, this is a mesenchymal stem cell based idea, right? So we are yet to see. It's going to be stored in liquid nitrogen for 20 years and they are just paying the rent for that. So unless they are really going to have a huge issue and its cells are thawed well and they are revived well. So I don't think so. We have any proper proof on that. Yeah, Pavitra, go ahead.
I was just going to say that there is no evidence, but lack of evidence actually supports the whole theory that maybe yes. So that is the...
Yeah.
So there is an alternate way. They can be used to, of course, later on convert them to pluripotent cells and go through the process. But for that, you don't necessarily need umbilical cord stem cells.
Any cell, yeah.
In fact, it was a good business sometime back. That's it.
Yeah, I think that answers the question. So next question is from Suviksha. How does cancer treatment based on cancer stem cells work? I don't know.
Cancer stem cell, okay, no. Cancer treatment based on cancer stem cell does not work. What we do is basically, you can identify how these cancer stem cells can be targeted or which population of those cancer stem cells can be targeted by specific drugs or what kind of regimen can actually substantially reduce your cancer stem cell population. And then you can identify this drug might actually work. That is the way it works.
Right, exactly, in personalized medicine.
Yes.
Okay, I guess that answers the question. So that's all from YouTube for now. So people who are in the MS team can raise their hand, and when they will be asked for them to put their question. Yeah, Yaswant Rao, you can unmute yourself and ask the question.
Then
Embryonic stem cells are said to be immortal or immortal, preferably. And cancerous cells also go on dividing forever without ending their lives. So what sets these two apart?
So there is a simple difference between cancer. In fact, this was an amazing question that prompted me to come to research. So way back in 2000, this was an amazing concept that was floating. So the difference is very simple. Both can perpetuate themselves forever, except that the embryonic stem cells can listen to you. That means you give them appropriate cues, they can stop self-renewing and make a decision to differentiate, whereas cancer cells never listen to you. Okay, that's the whole difference. Okay, that is what sets them apart. And molecularly, there are a lot of differences if you go look into the molecular aspects of it, but characteristic-wise, that's the major difference between these two.
Okay. Okay, next is Jayashree. Please unmute yourself and ask question. Hello. I guess he is not listening. So another question is from YouTube comment is that can we develop animal insect organoids using stem cells to avoid sacrificing them for production of drug, etc.?
Yes, that has been something which has been ongoing. In fact, more recently, there was, I think, organoids have been made from chimpanzees and orangutans, and brain organoids and spinal cord organoids have been made. So yeah, that is something which is very lucrative, but we should understand that organoids are still in their very primitive stage. Most of the stuff we want to make and to understand biology requires a properly formed vasculature, okay, blood vessels and blood system and, cues. So the external stimuli, the way we respond to external stimuli, those are not there in these organoids. So yes, they give you some kind of an advantage, but not necessarily at this stage. So until we have a thinking organoid, it is going to be a bit difficult.
Okay. Another question came in YouTube comment. Is that how can we use? Oh yeah, it's the same question as before for disease mapping. So someone is asking, can the panel member please address the controversy in the gene editing in babies? That's a controversy that means we can't.
This is designer babies actually. See, Chandrasekhar, what do you want to?
Yeah. Yeah, so basically, what is the limit we want to play God? That's the question. So one is, okay, there are two things. One is the moral question, the other is a technological question. Technologically, we still don't have the editing tools so well worked out or so efficient that they do not do what we don't want them to do. So they are not so precise yet. That's the technological issue, okay? So we still don't understand if we try to edit a gene, there could be many more genes that are modified to a certain extent, and the efficiencies are not still very great, okay? So the technology is still there, it is not there. That's one thing. You can get it, but you can't get it always right, because doing on a model organism is different than working on a human embryo. So you have to be like, you should have like sigma 6 perfection to do that, which I don't think the genome editing right now has it. That's one problem. Second is, to what extent do we want to modify? Now, this will also bring back a lot of issues from our history, that even long back in our history, we have decided to modify humans. Okay, the World War II was one of the reasons, this was one of the things that caused it. So, what is the extent we want to use it to editing in humans? That's a major moral question that is hanging around. It's not easy to. And how easily is it regulatable? Let's say even tomorrow we decide that this is the extent it can be allowed. How easy is it to regulate? Because the editing tools can be utilized in any IVF clinic very easily. So are there ways and means we can regulate it? So the general consensus as of now is that let's keep it off with respect to human germline editing. Germline editing means you make a change in the embryo before the gametes are formed. That means the mutation will now pass on to the next generation. There is another editing that is at the somatic level. You take the adult stem cells, change them and put them back, that's okay. Because that's going to be going off with that person. Whereas the germline editing will go to the next generation. Now that's a very serious business and we have to give a serious thought to it. So currently I think most of the scientists across the globe believe we should not get into the germline editing. Somatic editing is of course definitely acceptable, and that's what I think is permitted also as of now. So yeah, that's in a way what they did was a good thing. We at least became aware of the dangers that we are going to get in. Okay, you have a person who is not born and you are meddling with a person's genome. I don't think we have the rights to do it. So I think that's where it stands as of today. I don't think the germline editing would be permitted, and personally I feel it should not be. Okay, except in certain genetic disorders, I don't think it should be permitted at all.
Okay. So,
looks like Suresh has
connectivity issue is there. We have some connectivity issue. Yeah.
Yeah, you are fine now. You are audible.
Yeah. So Pawan has some question. I'm asking him to unmute himself. Okay, I think he's not listening.
Yeah, I'm here,
Yeah.
sir.
Okay.
Yeah, the previous question was mine, on which was addressed by sir, like keeping that in mind, I would like to ask to the entire panel, what would you think, or what would you hope would be the next frontier or the big development in the field of stem cell research in two aspects, research and technology, and the other aspect being how to bring down the cost of stem cell therapies. So could you address that from each of your perspectives?
Okay. Cost wise, let me just address the economic thing first. So the more it's like the bigger the market, okay, lesser your price, because you know that when TV was selling initially when it was first launched versus what happens now. I mean, I forget about the LED TVs and stuff, but otherwise I'm saying. So I think that is what. So if you have a bigger market, I'm sure that there's going to be a bulk production, which cannot happen, of course, with stem cell, because it is going to be, when you say that stem cell therapy, you're more often than not going to refer to a tailor-made therapy for that particular person. Okay, so the cost I can see marginally going down, but I think it's going to be a bit difficult. Probably in the next decade, there might be some kind of a miracle. And the next technology I foresee, which might alter it, is, I think we are already seeing the start of that. So this 3D printed organs, artificial organs. So you just have a scaffold and you embed your tissues in them, and you start seeing embed stem cells, and you start seeing different kind of 3D printed organs. So I think that's coming up in a big way. And similarly, you have this organs on a chip. So these kind of things are already revolutionizing the field. And from my perspective, a 3D culture of these stem cells and getting them as close as possible to organ systems, I think that would be really great boon for all of us.
Yeah.
Okay, so on the other side of the embryonic stem cells, in the past 3-4 years, there has been a huge shift in the field that's using the stem cells to model the embryonic development itself. So there were other models of it, like the embryoid body and all. They are very primitive. Now modeling means truly modeling the post-implantation development. Because in case of the embryonic development, even with respect to the human, you can only study till the embryo implants, because till then you can culture it in a dish. After the embryo implants into the uterus, you can't study it. You can use an animal model, but animal model will not tell you much because there is a huge difference in the development. So a couple of major groups working in Rockefeller and Cambridge have come up with methods to culture the embryos after the implantation stage. And people have been able to culture the human embryos till 14 days. This is going to revolutionize and give us better insights into the human development in the early stages. Now, there are certain ethical considerations. That's why it's not permitted to culture it beyond a certain time point. But I think there will be leniencies created in due course of time because it seems to hold great promise for understanding the human development, particularly in the early post-implantation stages. That's one field I think is going to pick up heavily in future. There are few labs around the globe working right now, and I think in due course of time there will be many more joining in because this will provide a great opportunity to understand the development as well as for understanding the developmental disorders.
Okay. So we'll be taking only few questions because of the time constraint. So another few among those few questions, this is an interesting one. Which therapy is better, stem cells or CRISPR editing? So what is your opinion, Dr. Chandrasekhar?
It is contextual. Basically,
Yeah.
see, if it's a degenerative disorder in some cases, okay, you can pump in more cells and do it. For example, let's say the corneal defect, okay, or we all know that LV Prasad Eye Institute is one of the great places doing the stem cell therapy. Now there is no question of using editing there. So it depends on what we are doing. So in such sort of therapies, I don't think editing is required at all. But there are certain congenital diseases or genetic disorders. Let's say, for example, sickle cell anemia, beta thalassemia. So the single gene disorders, mostly that's where the gene therapy, the genome editing may play an important role. But again, remember that there are two types here, in situ and ex situ. That is, in situ is, do you want to correct the mutation in the patient itself? Now, this prevents the problem of bringing it and getting more mutations. So there are no tools very well developed for that. Ex situ is mostly the editing, how it is done now. You take out the stem cells, edit them in the culture, then put them back. But again, as I said, once it comes back to the culture, it's considered to be a drug. It goes through a very different the QC is different as well as the procedures that are to be followed are very different. So I think it's still a long way, but as I said, it's contextual.
Okay. Another question is, what are your opinions about stem cells, use of stem cells to form meat production industrially? Dr. Devan, would you like to answer?
Yeah, sure. So, I mean, there's a lot of noise going about, conservation of natural resources because we do spend a lot in animal rearing and livestocking, right? So, considering several environmental issues, now this concept has come up, and of course, in CCMB we do a DBT-based project on this, where it's simply based on the principle that you isolate the muscle stem cells, allow them to retain the stemness for some period, and then make progenitor cells, and take it to the routine route of proliferation and then differentiation. So this way we are going to reduce the slaughtering of animals. But it, of course, the concept is quite rosy, but the challenges would be about scaling up and what all ingredients you would require to keep up the cell cultures in a bioreactor format, so that you have ample number of cells to make lab grown cultured meat. In addition to that, there are other cells which are involved, like adipose tissues, I mean adipose cells, and you need to bring in some vascularization. So they all together make meat, right? So all these experimental challenges are there, still technical challenges are there, but it's possible that based on the stem cell idea and the principle, we can do something which is it's going to revolutionize the whole environmental debate that's on.
Okay, that gives us a very good answer. So we won't be able to take any more question in this discussion. So Dr. Pavitra, are you with us?
Yeah, I'm here.
Okay. So I would at the end I would like to thank all the panel members for their ideas and opinions about all the questions that have been asked, and I personally learned a lot from this conversation that I didn't know before. So this gives us a very good idea for the general public. It gives us a very good idea that what stem cells are, how they can be used, what are the ethics around it, and also I think this has motivated many people also in the young minds to come and get into the research field of stem cells. So that's all from the panel members. And
thanks a lot, because we've had some really interesting questions and some interesting discussions. Thank you very much.
And for the audience, please fill the feedback form for the session. And also, I would like to give some information about tomorrow's session. The first session is also related to the stem cell research. That's the important. So people who are interested, please tune in at 10 o'clock at morning. The topic is brain evolution and organoids. After that, there we'll be having session about the different facilities that we have in CCMB. That includes cell culture facilities, that microscopic facilities, and zebrafish facilities, fly lab facilities. And also in the evening, we have a very exciting science quiz. So please participate in that. So that's all for today. I would like to thank again all the panel members for their time. Okay.
Thank you.
Thank you.
Thank you very much.
Thank you.
Thank you, sir.